Bismillahir Rahmanir Rahim. As we all know, interleukin-17 belongs to the interleukin-17 family, a group of pro-inflammatory cytokines. Interleukin-17 is vital for immune responses, tissue repair, and disease pathogenesis. Interleukin-17 works like an orchestra conductor, orchestrating immune responses, inflammation, and defense mechanisms. I have already made several videos about different rules done by interleukin-17, so you can watch it and understand how is interleukin-17 important in health and disease. My video today is about the interesting role of interleukin-17, which is the role of interleukin-17 in our metabolism. I'm Naglaa Elmongy, professor of dermatology, Cairo University. Here is my reference in this video, the metabolism modulating activity of interleukin-17 signaling in health and disease, which was published in Journal of Experimental Medicine 2021, May 2021. Interleukin-17 is best known as a protective cytokine in mucosal barriers, vital for immunity against fungi and extracellular pathogens, and also promotes inflammation in various autoimmune diseases. Interleukin-17A, the first member of interleukin-17 family, is mentioned here as interleukin-17. Interleukin-17 family includes other 5 cytokines, which are interleukin-17B, C, D, E, F. The metabolism modulating activity of interleukin-17 represents a newly recognized aspect of its functions. There is a growing understanding of the close relationship between the immune and metabolic systems, particularly evident in metabolic syndrome research, linking inflammatory cytokines from adipose tissue to insulin resistance and related disorders. Also, the metabolism of immune cells themselves is highly regulated. Immunometabolism refers to two interconnected aspects. The first involves how metabolic regulation within immune cells affects their behavior and outcomes, such as activation, proliferation, and differentiation. The second aspect examines how inflammation and immune responses affect overall systemic metabolism, including processes like adipogenesis. Interleukin-17 is transiently expressed during immune response to infection, but persists in chronic inflammatory and autoimmune diseases, where its impact on metabolic syndrome can be significant. In both contexts, growing research highlights interleukin-17's role in cellular and organismal metabolism. At the cellular level, interleukin-17-driven metabolic regulation supports cell proliferation in epithelial cells, keratinocytes, stem cells, aiding tissue repair and wound healing. Also, interleukin-17 enhances the proliferation and survival of synovial fibroblasts and fibroblastic reticular cells, facilitating interleukin-17-mediated inflammation in conditions such as arthritis and multiple sclerosis. And in addition to tissue repair, interleukin-17 plays a role in organismal metabolic regulation of adipogenesis, thermogenesis, and also on metabolic diseases, fibrosis, and tumorigenesis in disease situation. To get an idea about the immunometabolic response, we have to know that the immune system must quickly react to harmful triggers and adapt to changing environments, which requires readily available energy sources. And also to avoid the harmful effects of metabolites, cells maintain a balance between catabolism and anabolism, dependent on metabolic substrates like glucose, fatty acids, and amino acids. Pro-inflammatory stimuli often activate anabolic pathways such as glycolysis and fatty acid synthesis, leading to cell proliferation and differentiation. On the contrary, anti-inflammatory signals stimulate catabolic pathways like fatty acid oxidation, enhancing cellular quiescence. Also, from the other side, the metabolic state of immune cells determines the cytokines they produce and the resulting immune response. For example, M1 macrophages predominantly utilize glycolysis, while M2 macrophages rely on the oxidative phosphorylation. As we know, M1 is a pro-inflammatory, while M2 is an anti-inflammatory type of macrophages. A recent study found that gamma delta T cell subsets have unique metabolic signatures affected by the host's obesity status, showing that metabolic needs critically determine cell fate and function. As regards the immunometabolism in the interleukin-17 pathway, The sensitivity of interleukin-17 target cells is primarily based on the limited expression of the essential interleukin-17 receptor C subunit. This interleukin-17 receptor C subunit is typically expressed at low or undetectable levels in hematopoietic cells. Cells responsive to interleukin-17 are found in a variety of tissues and organs, each with unique metabolic and nutrient requirements. So the effects of interleukin-17 are context-dependent, resulting in variable physiological and pathological outcomes and occasionally contradictory metabolic regulations mediated by interleukin-17. Interleukin-17 signaling outcomes are also shaped by concurrent signals from other cytokines in the same environment. These factors contribute to the pleiotropic effects of interleukin-17 on both local and systemic metabolism. Now let's talk about interleukin-17 mediated metabolic regulation in barrier tissue. Interleukin-17 plays a key role in metabolically active barrier tissue repair as observed in the gut. This was highlighted by worsening symptoms in inflammatory bowel disease patients during clinical trials using anti-interleukin-17A interleukin-17 receptor A biologics for Crohn's disease, suggesting interleukin-17 regenerative importance. Interleukin-17 also helps skin wound healing, with studies showing that reduced interleukin-17 in murine skin injury models leads to delayed wound closure and compromised barrier integrity and skin microbiome dysbiosis. Interleukin-17 facilitates wound healing through multiple mechanisms. It stimulates the proliferation of keratinocytes and epithelial cells via the what's called ERK pathway and induces the expression of antimicrobial peptides like beta defensins and metalloreductase STIB4, which further promotes cell proliferation. The ERK or ERK pathway is a cellular signaling pathway that transmits signals from the cell surface receptors to the DNA in the nucleus. It controls various cellular functions such as growth, development, division, differentiation, and survival, and is frequently dysregulated in human cancers. Interleukin-17 also controls the positioning of the tight junction protein occluidin, responsible for gut epithelial barrier function. Its tissue repair effects are enhanced by other local cytokines, especially when synergizing with fibroblast growth factor 2 to activate genes that repair epithelial damage. The effectiveness of interleukin-17 induced repair might also be affected by nutrient availability. So, interleukin-17 signaling promotes cell proliferation and tissue repair, but when dysregulated, it can lead to tumorigenesis, particularly noted in chronic inflammation-related colorectal cancer. Interleukin-17 and tumor necrosis factor alpha synergetically activate NF-kappa B and hypoxia-inducible factor 1 alpha dependent glycolytic pathway, enhancing survival and proliferation in human colorectal cancer cells. A similar link between inflammation and tumorigenesis is observed in skin stem cells under interleukin-17 signaling. In this scenario, interleukin-17 facilitates the recruitment of the epidermal growth factor receptor to the interleukin-17 receptor complex in skin stem cells. This triggers epidermal growth factor receptor phosphorylation and activates the ERK5 pathway, leading to cell proliferation and potential tumorigenesis from wound healing. Thus, the interaction between interleukin-17 and epidermal growth factor receptor signaling in skin stem cells directly links growth factor signaling with tissue repair and tumorigenesis. As regards interleukin-17 mediated metabolic regulation in non-barrier tissue like adipogenesis, it has been observed that mice deficient in interleukin-17 receptor or interleukin-17A tend to be overweight and accumulate fat as they age. Interleukin-17 limits adipogenesis via transcription factors that control adipocyte differentiation, suggesting that the absence of interleukin-17 creates conditions in favor of adipogenesis. Interleukin-17 plays a role in regulating glucose metabolism with effects that vary depending on the context. Interleukin-17 plays a role in the development of insulin resistance by mediating immune responses and stimulating the production of other pro-inflammatory cytokines like interleukin-6 and especially tumor necrosis factor alpha, which contribute to insulin resistance. Actually, the field of immunometabolism is complex, and the precise ways in which interleukin 17 impacts insulin resistance are still under study. Now let's talk about another function of interleukin 17 mediated metabolic regulation in non-barrier tissue, which is energy homeostasis. Adaptive thermogenesis, primarily occurring in brown adipose tissue, which involves heat production, is facilitated by interleukin 17. This process is done by what's called uncoupling protein 1, which decouples mitochondrial fatty acid oxidation from ATP production, converting energy from substrate oxidation directly into heat, not ATP. Additionally, brown adipose tissue also is densely innervated by sympathetic nerves that release norepinephrine in response to stimuli like cold, activating lipolysis and thermogenic regulation. A homeostatic population of interleukin 17 secreting gamma delta T cells in adipose tissue is essential for maintaining body temperature. Interleukin 17 and tumor necrosis factor alpha signaling in brown adipose tissue stimulate the expression of uncoupling protein 1, facilitating adaptive thermogenesis. A study also indicated that interleukin 17F rather than interleukin 17A influences thermogenesis. Interleukin 17F is thought to enhance the expression of transforming growth factor beta 1 in adipocytes, which supports the sympathetic innervation of adipose tissue, thereby activating lipolysis and thermogenesis through norepinephrine. As regard the role of interleukin 17 in metabolic immunological coordination, interleukin 17 facilitates metabolic immune coordination in adipose tissue, and in aging mice, the population of interleukin 17 producing gamma delta T cells and regulatory T cells in adipose tissue increases simultaneously. Consequently, mice lacking interleukin 17A show a reduction in Treg numbers, in fact, indicating that interleukin 17 has a role in the homeostatic expansion of Tregs. And while the studies did not specify the impact of interleukin 17 mediated Treg accumulation on insulin sensitivity, Treg cells are recognized for enhancing insulin sensitivity. A critical issue arising from research on interleukin 17 in adipose tissue concerns its effect on human obesity and related metabolic disorders like diabetes. It's hypothesized that the increased interleukin 17 production seen in metabolic syndrome might represent the immune system's effort to counteract the metabolic disturbances linked to elevated fat storage and insulin resistance. Once obesity is established, the potentially beneficial metabolic effects of interleukin 17 can be lost by harmful mechanisms such as elevated tumor necrosis factor alpha levels, shifting the balance towards inflammation and exacerbating obesity-related metabolic complications like insulin resistance. Obesity-related metabolic diseases like type 2 diabetes and cardiovascular conditions are linked to chronic low-grade inflammation. And as I said before, the field of immunometabolism is complex, and the precise way in which interleukin 17 impacts insulin resistance are still under study. As regard the role of interleukin 17 in cellular immunometabolism during inflammation, autoimmune diseases like psoriasis and rheumatoid arthritis often affect the cardiovascular system, but the underlying mechanisms are mostly unclear. Recent research offers some insights. For example, in experimental psoriasis induced by imiquimod mode, interleukin 17 was found to increase collagen deposition in both psoriatic lesions and distal skin areas, as well as arteries. However, a recent time control study indicated that ustekinumab, a blocker of interleukin 12 interleukin 23p40 subunit, might trigger severe cardiovascular events in high-risk patients. Also, low interleukin 17 serum levels were linked to an increased risk of major cardiovascular events in patients with myocardial infarction. Also, in a mouse model of hypercholesterolemic atherosclerosis, interleukin 17 administration decreased endothelial VCAM1 expression, vascular T cell infiltration, and atherosclerotic lesion development. However, the circumstances in which interleukin 17 acts as a pro-atherogenic or protective remain unclear, suggesting that careful monitoring is required when using anti-interleukin 17 therapy in patients with cardiovascular risk factors. As regard the role of interleukin 17 in fibrosis resulting from inflammation, various fibroblasts and mesenchymal cell types across multiple organs, regulate extracellular matrix, homeostasis, and like other cytokines such as tumor necrosis factor alpha and transforming growth factor beta, interleukin 17 can mediate fibrosis following a dysregulated healing process. Interleukin 17 is associated with collagen production in conditions like angiotensin-induced hypertension, bleomycin-induced lung fibrosis, and skin fibrosis. Also, blocking interleukin 17 can reduce tissue injury, inflammation, and fibrosis. However, in a kidney injury model, interleukin 17 acts as an antifibrotic agent by stimulating bradykinin production through the kallikrein-kinin system in renal epithelial cells, leading to the generation of matrix metalloproteinases that limit fibrosis. Thus, interleukin 17 role in tissue remodeling is shaped by the cell type and the local inflammatory context. So, as a conclusion, interleukin 17 is more than just a pro-inflammatory cytokine. Research indicates its significant role in regulating metabolism at cellular, tissue, and organismal levels. Additionally, cellular metabolism can affect interleukin 17 signal transduction. During inflammation, a feed-forward loop is initiated where effector molecules from interleukin 17 target cells influence systemic metabolism, which in turn affects the metabolism and function of these target cells. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel. And thank you.